Welcome to the virtual soybean disease field tour. Thank you for attending. I hope to give you as good as a tour as I can virtually. This first part we're going to talk about is disease identification. And so as one of the handouts you can use to follow along, it lists the major soybean diseases I'm going to be talking about. It gives you a spot to fill in symptoms under foliar symptoms, talking about the leaves, stem symptoms, and root symptoms. I also want to let you know that where we're doing this is in the variety trials. What I do is I take the varieties that have been entered in the county standardized trials for University of Tennessee. I put them in small, randomized, replicated trials. And this is one of 2 to 3 locations. I look at these varieties. They're replicated 4 times at each of these sites. And at each of these sites, they're 4-row plots. The right 2 rows of each plot have been treated with a fungicide. This year's Quadris Top SBX at 7 fluid ounces, treated at growth stage R3. And so as we're walking, you can keep that in mind. The sign for each variety is in the middle of the plot. So the left 2 rows have been untreated or are not treated, and the right 2 rows are treated. And so they're grouped by maturity group 3, early 4s, late 4s, and then maturity group 5s. And then our Liberty links that are early 4s and late 4s are in a separate area on the other side of the alleyway here that we'll also be walking. And so keep that in mind. All of these varieties I rate for disease, as well as we take them to yield. And all of that information can be found at search.utcrops.com. So again, one of the first questions I then tell someone in response to their questions, should I put a fungicide out, is what's your variety and what are you seeing? And so here I hope to give you the information to identify what you're seeing, and then you can go to search.utcrops.com to look up your variety, and there you can possibly see an idea of what response you might see to a foliar fungicide in your variety if we've been able to test it. So the first major disease I'm going to talk about here is frog eye leaf spot. Still one of the major foliar diseases in Tennessee, if not across all soybean growing states. But it's very highly dependent upon the variety you are after, or the variety you plant. This variety is in our border rows here. It gets a really good frog eye leaf spot. So foliar symptoms for frog eye leaf spot: you're going to get a lesion on the leaf. It doesn't always have to be perfectly circular. It can be slightly angular. The big thing is you're going to have a dark border around that lesion, dark brown to purple. And you want to flip that leaf over and look at the underside. So you have a lesion with a dark border, and on the underside of that leaf, in the center of that lesion, you should see a dark spot. So that kind of gives it that frog eye appearance. And the reason we call it frog eye leaf spot. That center dot is the fungus sporulating, Cercospora sogina. And so again, foliar symptom, lesion, dark border, underside of that leaf on that lesion, a center dot to show it's sporulating, because you can get injury from chemicals that look similar to this, but that lesion will be more bleached out in the center, and it won't have that dot on the underside of the leaf to show sporulation. Again, placement in the canopy too. Majority of the frog eye leaf spot will be in the upper canopy. Not to say that you don't get it throughout, but you can see the majority of your lesions are going to be more in your upper canopy. And so that's important to note as well as we talk about some other foliar diseases. Frog eye leaf spot can cause lesions on the stem and the pods, but it's not as frequent as you would see on the leaves. You really have to have a high infection or very susceptible variety usually with the right weather conditions. And you don't have any root symptoms associated with frog eye leaf spot. Any questions about frog eye leaf spot? While this one can be managed with variety, fungicides work very well, and we'll talk about that later on also. But we do have resistance to the QOI or the strobilurin class of fungicides in frog eye leaf spot. And so we have continuously for multiple years now, we don't recommend going with a solo fungicide product that just has that one active ingredient that's in that strobilurin or that QOY class. And that means on that fungicide label, it would only have an 11 on that label. You want to make sure if you're doing that, you're pairing it with another fungicide that's got a different number on the label, usually say a trizol or a DMI, which is a number 3. So we're going to move into the alleyway here and walk down between some of our varieties to point out some additional diseases. So here you have a 3. You can see again, these 2 rows have been treated with a fungicide, slightly greener. The 2 rows that have not been treated with a fungicide, starting to turn a little earlier. When you pull the canopy back, this is when you can see some defoliation, of course, already starting, as well as target spot. So target spot's the next disease I'm talking about. This is a foliar disease. You're going to see lesions in the lower canopy. And here, the lesions can be bigger in size. It doesn't mean they don't start smaller, but they're a solid, usually solid brown color. They may or may not have concentric rings, and they may or may not also have a halo associated with them. But the main thing is they are a solid brown lesion in the lower canopy. And they can cause defoliation in the lower canopy. Here too, they can cause some lesions on the stems and the pods. But again, the majority of the symptomology to identify it would be on the leaves in that lower canopy, and no root associated symptoms for target spot. It can be mixed in a lot of times with septoria brown spot. And as you'll hear from Ty Smith, my PhD student, on studies looking at fungicides and also screening it for fungicide resistance. Again, here you can see the difference in just protecting the overall foliage using a fungicide, but you can pull the canopy back and you still see target spot in a treated area. This is where you might see the smaller target spot lesions, again, that are very same size as frog eye leaf spot, but they're solid in the color, that the solid color, slightly yellow halo around them. And again, that they're in the lower canopy. But it's interesting because you might say, oh, well, there's more lesions maybe where it's been treated. Well, it's mainly usually because there's more leaves still on that lower canopy to find lesions versus where it's not been treated. Those lower canopy leaves may have already fallen off, so you don't see those lesions. Any questions about target spot? And for those of you watching virtually, always feel free to email youngkelly@utk.edu with any questions, or put them on the comment box within the news.utcrops.com article that this is posted in. All right, so I'm going to ask for a little bit of participation from the in-person audience here. Where this flag is at, we've got one plant showing definitely something going on. We've got interveinal yellowing, which is, we call interveinal chlorosis, even necrosis where it's dying. Who wants to just take a stab at it?
Is it stem canker?
Stem canker or SDS? So that's a really great question, and that's exactly what I wanted to hear. The other thing too, I want to point out that newest growth, you look at the smallest leaf on the plant, still showing these symptoms, indicating we're not dealing with injury, as well as it's just one plant. It's not across the entire plot. So something you always want to take into consideration to make sure you're not looking at just a injury initially. You're seeing it in the newest growth. Definitely the foliar symptoms of both stem canker and SDS, which I'm going to kind of talk about them both at this stop. The foliar symptoms are pretty much exactly the same between the two. It's very hard to tell them apart just based on foliar symptoms. You've got that interveinal yellowing or chlorosis that can turn to necrosis, brown dead tissue between the veins. One thing that isn't always, it doesn't hold 100% true, but a lot of times with stem canker, as the leaves die, they'll stick onto the plant. They won't fall off. versus SDS or sudden death syndrome, the leaves will fall off and you'll just have petioles left sticking out of the plant. That's not to say you can't have a mix of both, but when you have a lot of either sudden death syndrome or stem canker in a field, and later on in the season, that might help you differentiate it if it's past the point of being able to use some of the other characteristics. So now we're going to move to the stem. And so here, you always want to pull that canopy back, look at both sides of the stem, and here you can see a nice dark canker or lesion on that stem. It's kind of that first true node up from the soil line, and it's only on this side of the stem, one side, not girdling around the entire stem. And so that indicates right then that's a stem canker. So as the name suggests, which is really nice, you should find a canker on the stem, and southern stem canker will just be on one side. And so with an SDS, a sudden death syndrome plant, you're not going to have that lesion or the canker on the stem. When you go to split that stem, as long as Dectes stem borer hasn't been in there, running up and down it, eating that center pith, turning it brown, you have not had Dectes in there, you can cut it open. That center pith should be brown, especially under that lesion for stem canker. So that's one of the stem things for stem canker is that center pith will be brown, where SDS, that center pith should be white. But the vascular tissue on the outside of that center pith will be discolored from SDS. So that's the difference there for the two stem symptomologies. And then when you go to pull up these plants, stem canker, it's still going to have a pretty good root system. You're going to have to put maybe a little bit of effort into pulling it out. Again, there's that canker just on one side, not the other. And so for roots, stem canker, you should still have a good root system. You don't have symptomology with stem canker. SDS, a lot of times you'll have a weaker root system. Not necessarily saying you won't have a taproot, but usually you'll have a weaker root system. That plant will be easier to pull out of the ground. Because what's going on is with sudden death syndrome, that fungus has actually colonized the root system earlier in the season, and the above ground symptoms you're seeing are the toxin that pathogen produces and goes into the vascular tissue up into the leaves for sudden death syndrome. And so that's the difference between the root system symptoms for stem canker and sudden death syndrome. Are there any questions about that? So with this one variety, in this one plot, I would note that there's stem canker in this variety. But I wouldn't report it as being susceptible to stem canker unless I find it in at least 2 of the 4 plots at that location. And I want to show you why, though. Participation from our audience here. We've again got this one plant, what looks to be just one plant, showing very similar symptoms, interveinal yellowing, some necrosis. What do we think it could be? I heard a whisper of stem canker, right? And so what do we need to do to check?
Look for the canker.
Look for the canker on the stem. That's right. So when we pull that canopy back, we find actually a dead plant next to it, but we do find that canker right there on one side of that skinny little stem. Right there under my finger. But when I see this dead plant right next to it, it's almost darkening from the soil line up. There's no leaves left on it, just petioles. In our area, this would actually tip me off that I might have a new disease here called taproot decline, which I will get to at the very end of our disease identification stop, where I have a really good plot that only has taproot decline in it. Again, the petioles sticking out without the leaves later in the season could indicate we had SDS here. But looking at the entire field or this entire plot of this variety, and knowing in my area, I don't usually see SDS do this. Different areas, you might see SDS do this. But really, Mid-South, Southeast, you would expect stem canker to almost hit plants and make them decline very quickly, versus here, sudden death syndrome isn't as quick as it is the further north you go. And really, sudden death syndrome, a lot of times you might just see a sprinkling of it in a field and you can still have really good yield. My predecessor, Melvin Newman, had a really nice saying, well, you know, sometimes you might even just want to see a little bit of SDS because it indicates a really good growing season. That same weather and growing season that the sudden death syndrome pathogen thrives on, so do the soybean plants. But it's very much going to be highly dependent upon your variety, how much SDS you have in the field, as well as if you have a secondary pathogen out there or pest, your soybean cyst nematode. So again, we highly recommend you soil sample, because if you have soybean cyst nematode, you need to be addressing and managing both soybean cyst nematode and SDS. If you're only going to manage SDS and you have soybean cyst nematode out there, you're going to really have a hard time, because those combined make the damage and yield loss from SDS much worse. So here's a variety, the 4 rows in front of me, and this is all stem canker out here. And so again, this same variety in a different spot of the field might only have one plant that's affected. But given if it's put in the right spot of the field, the right conditions with that pathogen present, stem canker, especially in the mid-south and southeast, is very, very devastating. And you may not know you have stem canker in your field. If you have had it hit your field, make sure you keep those records so you know never to go with a susceptible stem canker variety there, because it can definitely hit you hard. And again, it's individual plants, but it can be scattered throughout. That's a little bit of another trick, is distribution of symptoms in a field can help give you a handle on what you might be looking at. SDS, you'd kind of maybe expect it to start in one spot and expand out a little bit, maybe to the size of a truck area, and then you'd have hot spots in the field. And then again, stem canker could be similar, or again, it could be scattered throughout, but individual plants. It's going to, you know, again, vary from field to field to some extent, as well as what variety you're using. So this plot here, nice and clean upper leaves. You open up that canopy and pull it back. And what I liked about this variety was to show you, because it's pretty much just Septoria brown spot. It's easy to kind of identify diseases when that's the only thing you're looking at. And so this was a good plot to just show you Septoria brown spot. So again, this one's going to have foliar symptoms just in the lower canopy. Small specks. They're very small lesions, but they can coalesce to form larger lesions associated with yellowing or slight yellow halos with them. But again, definitely not near as formed lesions as the target spot. That's not to say, again, both of these can be present in the same field, so it can be hard to say whether you just have one or the other, or you a lot of times could have both. So far, all varieties are susceptible to Septoria brown spot. I've never found a variety that I've not seen Septoria brown spot in, but the level of Septoria brown spot can vary across varieties. So again, lower canopy disease, small speckled lesions that can coalesce into larger lesions with some yellowing associated with it. This is a disease you can see earliest in the season, even on seedlings, but beware, it can look very similar to herbicide injury. It is considered a secondary disease. It's never usually getting up out of that lower canopy, and it's usually not robbing much yield. But a lot of times, if we're seeing a response to a fungicide, and there's no other significant foliar pathogen that we're controlling, a lot of the times it could be contributed, the response and yield we see, to cleaning up the Septoria brown spot. Any questions about Septoria brown spot? Alrighty. Okay, here we're at another stop. Here we're talking about Cercospora leaf blight. And this is the showiest I could give you at this point in time. Given another week or two, this would be very showy. Virtual tour, I'm going to put up a picture of one that's progressed further along to really show that bronzing symptom. Sometimes those leaves will curl slightly when they start to get just slightly point down. but you get that bronzing or purpling on the leaf for Cercospora leaf blight. It's going to be in the upper canopy, and with that you have this symptom, the bronzing symptom, in the upper canopy. You can also have the blighting symptom. The blighting symptom is going to look very similar to almost like Septoria brown spot has gotten up to the upper canopy. They're going to be smaller specks, lesions, but they're going to be clustered on the edge of the leaf, almost like that leaf is a little burnt. So you have your blighting and your bronzing. Some really, really interesting research out of LSU with my colleagues there and their graduate students. They took bronzing symptoms and blighting symptoms, and they looked for the toxin, cercosporin, that the pathogen makes, as well as quantifying the amount of pathogen was present using molecular techniques. While they found both the blighting and the bronzing had the toxin present, the blighting symptom had the highest level of pathogen present, where you have very low levels of the pathogen present on most bronzing leaves. And so we're really much more concerned going forward with that blighting symptom of Cercospora leaf blight. As further investigations are still ongoing, we have regional work working on Cercospora leaf blight. Here in Tennessee, Cercospora leaf blight, I really only see it come in later in the season. I don't usually see it affecting yield, and it's coming in late enough in the season, I don't see a response to a fungicide. Again, further south you go, that changes. Louisiana, that's one of their #1 foliar pathogens down there. That again, we're working as a regional team to help look for varieties that are resistant to it, as well as if there's some consistent fungicides out there for Cercospora leaf blight. Not going to have any, you can get some symptoms later in the season again on the petioles for Cercospora leaf blight, and maybe even on the stem. But again, the majority of the symptomology is the bronzing or the blighting on the leaves, and no root symptoms with Cercospora leaf blight. Cercospora leaf blight, though, is the same pathogen that causes purple seed stain. But another internal study we did, my first full year on the job, poor undergraduate student, we opened up a bag of seed that we had received that actually was untreated. And we looked in it and we said, oh wow, there's a lot of purple seed stain in this. Let's put a trial together. Well, when you think there's a lot of purple seed stain in there, there's really not near as much as you think there is when you ask a student to go pick out every single seed that has purple on it. It was really a big task because she was scraping the bottom to really get enough seeds for a trial. But we did it, and again, we did a factorial test looking at seed treatments and foliar fungicides. Well, we didn't get any Cercospora leaf blight that developed in that trial, even though we were using purple seed stain seed. So just again, because you had it on the seed doesn't mean you're going to get the foliar disease. But the purple seed stain, usually the amount you have on the seed might affect germination and vigor. But again, for the trial we did and the seed treatments we used that year, we did not see a reduction in vigor or stand due to the purple seed stain. Again, I don't think we had probably a high enough infestation on that seed to be able to actually see an effect. But it is known, usually I think about 40 or over 40 and over 50% of the seed has to be covered with that purple color to actually be affected. And so most of the time, the seed companies are not letting those get through screening anyways, so you have good vigor and good germ seed. So in this exact same spot, now this is the 3rd year, I found taproot decline in this spot. 3 different varieties have been planted here. All varieties had a seed treatment on it. And so taproot decline, earlier in the season, you might just see a couple plants with this yellowing symptom. So the leaves are yellowing, but those veins are kind of staying green. Almost a spider web kind of vein look. So that's the foliar symptoms. And when you go to pull back the plant canopy, what you'll notice is there's dead plants there's plants that have died prematurely. So similarly, when I talked about, we found that 1 hit of stem canker, but when we pulled that canopy back and we looked in the row next to it, we found some plants that had died prematurely. And so similarly here, the best example I have for you is actually in a row next to the highly affected plants. You have a plant right there, died prematurely, didn't produce any pods. Definitely, no yield off of that plant. The other thing I'll show you virtually, when you look around move the leaf litter back, you're looking on the old debris stems or more roots of the previous crop, you might find dead man's fingers. These are these black protrusions coming up out of the previous debris covered in white spores. And at this site, I've actually never found that characteristic symptom, but we have confirmed this is taproot decline. The stem, again, as we saw in that other area, it just kind of dies from the soil level up. So it'll start to discolor from the bottom up and be darkened. And really, when you go to pull out a root or pull out a plant, the root system is very, very much weakened. A lot of times that taproot will break off in the soil. Sometimes you can see the growth of the fungus on the outside of the root, as well as if you split the stem in that center pith near the soil line and into the root, you might be able to see the fungal growth of the pathogen. And so again, a very new disease. Xylaria is the genus, is a brand new species. This the pathogen, that genus and the family is actually, the majority of it are wood rotting fungi. They're usually in the woods rotting trees that are dead. There are some examples, other examples of pathogenic species in that group. Black root rot of apple is one. And so molecular as well as morphological and management research is going into this pathogen. Again, hoping we'll find some varietal resistance, possibly maybe seed treatments, although again, I said the last, this being the third year we found it in this site. It's always just been the first plot of my Liberty Link variety trial. So it's been a different variety each year with a slightly different seed treatment, whatever comes on that variety. And we've continued to see it. The good thing is it hasn't spread very largely, if any at all. It's always been confined to 2 to 3 rows since we found it the first year at this site. But it's very important when you are identifying diseases, and if you identify a soil-borne disease in your field, that you're mindful about moving equipment from that field to other fields. You want to make sure you're cleaning it off, getting all the soil and or debris off of that equipment and not moving it to another site to then spread that disease. And so that's really one of the best management recommendations right now. Make sure you're IDing what you have if it's taproot decline, and make sure you're not moving it to other fields. We are continuously interested in this pathogen system, wanting to get and identify more locations to know our distribution. So far in Tennessee, we have identified it in Hardeman, Haywood, Madison, and Gibson County. And that's not to say it can't be in other counties. We've just not found it to ID it yet. I have a graduate student, Shelley Pate, who's starting to investigate it for Tennessee and specifically targeting to see if there's any synergism, interaction between nematodes and this soil pathogen. So again, we've talked about the yellowing with the green veins for the foliar symptoms, essentially earlier dying plants when you go to look at the stems, and that those plants have kind of died from the soil up, and that the root system is again greatly weakened, where a lot of the lateral roots and the taproot might actually fall off or not even come out of the soil when you pull up a plant. mix and match comparisons, what I do is my verification, what I call my verification trial each year. And for about the last 3 or 4 years, what I've done is I picked 3 varieties that differ in their susceptibility to frog eye leaf spot, as well as to other pathogens. And then with that, I pick 5 fungicides that I'll evaluate across those 3 varieties. And so that's what we'll walk here. So the first rep is in order. So our first variety is that moderately susceptible variety to frog eye leaf spot. Again, when I get out there and squat down in there, the frog eye will pop out at me more than it does just standing from the outside of the plot with the way the sun is right now. But when we get to the next variety, you'll see it's much easier to see the frog eye on it, and that's why it's more our fully susceptible variety. NTC is non-treated, check. And so then we'll start to walk through the 5 fungicides that we're evaluating, or mixtures we're evaluating this year. You've got Quadris or generic azoxystrobin plus Tilt or generic propiconazole. And what you'll see is across these first 2 varieties, all of our treatments reduce frog eye leaf spot. But when you look at averages of yield response across years and across these varieties, again, going with the more economical treatments, you usually don't get as big as a response in yield as compared to our newer products on the market or our pre-mix fungicides. The next one, again, Quadris or generic azoxystrobin, Tebustar or a tebuconazole product. What I want to point out here with tebuconazole products, this is one variety. You might look across and not see anything wrong. But if you actually walk the variety, look really, really closely back at the area where the leaves were out when it was sprayed, you actually see what you might think is sudden death syndrome. Again, you got that yellowing in between the veins, maybe even a little necrosis, dead tissue. But again, looking at that newest growth, you don't see any of that going on. All that newest growth is clean, indicating, go back to your records, what went out on this field. And it's actually an injury. And so again, this variety did not rate high for phytotoxicity, what we call that chemical injury, a phytotoxicity rating, but it varies across these 3 varieties. And across the years, I've had the same frog eye leaf spot resistant variety, and for one reason or another, that variety is more sensitive and does show phytotoxicity greater than other varieties, which has been seen before. My counterpart, Tom Allen in Mississippi, the further south you go, sometimes you can more regularly see this phytotoxicity, and he was able to put a graduate student looking at phytotoxicity in soybeans. But with that project, he did not see a significant or statistical yield loss to phytotoxicity. But again, it doesn't look good when you see it. Again, anywhere from 3 to 14 days after application, you can see phytotoxicity. Usually, how early you start scouting, you might just see a little bit, and then it does progress, hits its plateau, and then as new foliage comes out, it looks like it drops off in its severity, but it's really just you've got new green foliage that kind of covers it up or hides it. And so just to keep that in mind as we walk the other varieties with that treatment, to see the difference in phytotoxicity. Maravis Top, this is a new product on the market from Syngenta. It has their new SDHI component, adepidin in it, plus difenconazole. You can kind of think of this as the new Quadris Top. So the difenconazole component is the same that's in the Quadris Top, but they've kicked out the azoxystrobin and paired it with the SDHI adepidin. It's been looking very good. As Ty Smith will also talk about it in target spot trials. And then we have Revitec. This is the new product from BASF. It's a 3-way. It's got their new DMI component in it called Revasol. So you can think of this as the new and improved Preaxor. So Previously, we looked at, Preaxar D, which was Preaxar plus Domark, or Preaxar plus Tilt. Well, they've now taken Preaxar and paired it with their own DMI called Revysol for Revitech. This product has also been looking very, very good, keeping the beans very clean, and again showing that yield increase as we've continued to see that consistently in our top fungicide products. The last product I threw in this trial is Acropolis. So it actually does not have a QOI in it. It is our Topsin, which is thiophanate methyl and MBC, and tetraconazole, which is the product, the AI in Domark. So a strong triazole with Topsin. Topsin is look good, but it has a high risk for fungicide resistance development. So I like to see Topsin paired with something. Here it's paired with a good triazole. I do like bumping the rate to 23 ounces and not just the 20, but it hasn't always given me as consistent results in some of the other pre-mixed fungicides. And so we now get into our next variety. Again, here's our fully susceptible frog eye leaf spot variety. Here the frog eye pops out at you more. It kind of hits you right in the face. And so here again, you can pick out more easily sometimes differences between fungicide products when you have more disease pressure. Again, a lot of times if you go across your field and you spray your entire field with a fungicide, it's hard for you to say, is it really doing me any good? And that's why it is nice to show you at field days or virtually the difference between untreated here and a fungicide treated plot. And again, all the fungicides on the market are definitely going to help. And so even here, again, a generic azoxystrobin, propiconazole, 6 fluid ounces and 4 fluid ounces, you see a significant reduction in the amount of frog eye leaf spot. But again, showing you the data across years helps you understand though why we want multiple years of data. We also do this at up to three different locations per year to show different disease pressures. So you can get an idea of what's going on, because it's going to depend on your variety. It's going to depend on your field location, what kind of response you might expect in respect to yield. Again, here's that Quadris plus tebuconazole product. Again, you have to really look in here and look back to that level of when it was sprayed to try to find phytotoxicity. But again, here, not near as much as what we saw in some of the other, that other variety, but also a little bit harder to see right now. It's not near as showy as it was just a couple weeks ago. Again, all these applications went out at growth stage R3. Again, here's your Meravis top. Very, very clean, just as your Revitech plot is too. With your Quadris plus your Tilt, or your Quadris plus your tebuconazole, you still saw some frog eye peeking through there, where the last two treatments there, really nice and clean. Your Acropolis here, very clean also, maybe with just a couple spots showing through. Again, you can see the ratings on the table and or graph. And then the last part of the trial I'll walk you through is our resistant variety. Again, when we say resistant, it doesn't mean that you might not find a lesion of frog eye in it. It just means respective and comparative to other varieties, it gets the lowest amount of frog eye. And so very clean. So again, here you're not going to see visually a lot of differences, if any at all, between the non-treated check and our fungicide treatments. But here where I have my last flag marked, even from a distance, maybe with the way the light is shining, you can see already there's much more phytotoxicity, as I mentioned, in this last variety compared to others. So again, focusing like maybe right there, you can really see it. Here even, the phytotoxicity got bad enough where we even had some necrosis, not just the yellowing. So again, yes, question. We did. We spray this with a spider rig, so we can adjust the height of it. Again, it spans all 4 rows We treat all 4 rows the same, but collect all our data out of the center too.